Hi, I'm Ryan O'Mara, a PhD student from the University of Ottawa and Ottawa Hospital Research Institute. This protocol will describe methods on how to produce enriched populations of oligodendrocyte precursor cells from mice, which may be differentiated alone or in combination with mouse dorsal root ganglion neurons to produce myelinating co-cultures. Let's begin. To give a brief overview of the procedure, neonatal mouse cortices are dissociated and seeded into T25 flasks. After a culture period of 9 days, an astrocyte monolayer forms on the base of the flask, on top of which oligodendrocyte precursor cells are proliferating. Oligodendrocyte precursor cells are then obtained and purified and can be subsequently differentiated into mature oligodendrocytes in an enriched culture. If myelinating co-cultures are desired, dorsal root ganglion neurons from P5 to P10 mice are cultured alongside the mixed glial cultures. Over a period of 9 days, these neurons form a dense neurite bed on which oligodendrocyte precursor cells may be seeded. This allows for oligodendrocyte-mediated wrapping of the neurites, thus resulting in a myelinating co-culture. First, whole brains from P0 to P2 mice are placed in ice-cold MEM and transferred to a dissection microscope. Using a scalpel, cut through the meningeal layer of each cortex in a longitudinal manner. Then pry off the meninges from the underlying cortex. Next, remove the olfactory bulbs. With the brain ventral side up, make deep longitudinal incisions where the cortices meet the ventral diencephalon. With the brain dorsal side up, pry off the cortices. Then dice the cortices into about 4 pieces each. Transfer the cortices into a 15 milliliter conical tube containing 350 microliters of ice-cold MEM per brain. Brains of the same genotype are pooled in the same tube, so that 4 brains would require 1400 microliters of MEM. Once the dissection is completed, move the pooled brains to a 37-degree water bath for 3 minutes. Next, in a sterile tissue culture hood, pass the brain pieces through a P1000 pipette tip to break them up into smaller pieces. Add 75 microliters of pre-warmed OPC papain per brain to the conical tube. Then incubate in a 37-degree water bath for 20 minutes, inverting the tube every 2 minutes to prevent tissue aggregation. Once the incubation is completed, add 2 milliliters of mixed glial culture media per brain to the digest. Allow to stand for 10 minutes. After 10 minutes, aliquot the tissue suspension into 5 milliliter plastic tubes. The number of tubes should match the number of brains in the preparation. Using a flame-polished glass Pasteur pipette, triturate the contents of each tube slowly at first, then with increasing intensity as pieces begin to dissociate. Stop triturating once there are no visible chunks of tissue left in the suspension. Pool the contents of each tube into a 50 milliliter conical tube containing 4 milliliters of mixed glial culture media per brain. After gently inverting the tube several times, aliquot the cell suspension into 15 milliliter conical tubes. The number of tubes should match the number of brains in the preparation. Centrifuge the tubes at 300 times gravity for 5 minutes. and resuspend the pellets in 1 milliliter of mixed glial culture media. Add the resuspended cells from each conical tube to polyal-lysine-coated T25 flasks that were pre-equilibrated with 5 milliliters of mixed glial culture media. The number of flasks should equal the number of brains dissected. Incubate the flasks in a 37-degree incubator at 8.5% CO2. After 3 to 4 hours, the cells should be adhered to the substrate. However, there will be much debris persisting in the media which may adversely affect cell viability. To avoid this, replace the media after 3 to 4 hours with fresh mixed glial culture media. As you can see, this media change removes much of the cellular debris. The next day, many cells should be evident on the base of the flask. On the third day, an astrocyte monolayer should be forming on the base of the flask. At this point, perform a 2/3 media change by removing 4 milliliters of media and adding back 4 milliliters of fresh media. This step should be repeated every 3 days. On the 6th day, an astrocyte monolayer should be clearly visible, on top of which oligodendrocyte precursor cells are proliferating. At this point, supplement the culture media with insulin at a final concentration of 5 micrograms per milliliter. On the 9th day, the oligodendrocyte precursor cells are ready to be separated from the astrocyte monolayer. To do this, place the flasks on an orbital shaker in a 5% CO2 incubator. Allow the flasks to equilibrate to this incubator for 1 hour. Then, shake the cells at 50 RPM for 45 minutes. This step removes many loosely attached contaminating cells. Perform a full media change by removing all of the media and adding back 4 milliliters of fresh culture media. Replace the flasks in a 5% CO2 incubator and allow to equilibrate for 3 hours. At the 3-hour mark, check the coloration of the culture media. The culture media should be red, indicating a balanced pH. If the color is towards orange or purple, adjust the incubator's CO2 level accordingly. Maintenance of a balanced pH is important for the survival of the oligodendrocyte precursor cells during the overnight shake. Once a balanced pH has been achieved, shake the flasks at 220 RPM for approximately 16 hours. The next morning, remove the flasks from the shaker and transfer the media from the flasks to 10 centimeter tissue culture dishes. These dishes will serve as means to further purify the oligodendrocyte precursor cells by differential adhesion. Each dish should receive media from 2 flasks. Incubate the dishes at 5% CO2 for 30 minutes. At the 15-minute mark, give each dish a gentle nudge to prevent oligodendrocyte precursor cells from adhering to the plastic. After the 30-minute incubation, examine the dishes under phase contrast microscopy. Oligodendrocyte precursor cells are evident as small clumps of cells. Some of these cells may have adhered to the dish. Loosen the adhered oligodendrocyte precursor cells from the dish by gentle agitation. Be careful, too much agitation may result in loosening of the contaminating cell types. Transfer the cell suspension to a 15 milliliter conical tube. And centrifuge at 300 times gravity for 5 minutes. Resuspend the pellet gently with a P1000 pipette tip, followed by a P200 pipette tip. Count the cells with a hemocytometer. Purified oligodendrocytes may then be seeded onto laminin-coated cover slips for differentiation into enriched oligodendrocyte cultures. At day 1 post-seeding, the enriched oligodendrocyte precursor cells have relatively simple morphology, extending only a few processes. By day 3, differentiating oligodendrocytes have extended numerous processes reminiscent of immature oligodendrocytes. By day 6, oligodendrocytes have extended myelin-like membranous sheets. At any point during this time course, cover slips can be fixed and processed for immunocytochemistry. Rather than seeding the purified oligodendrocyte precursor cells on laminin-coated cover slips, they may be seeded onto dorsal root ganglion neurite beds to produce myelinating co-cultures. This next section will describe methods on how to produce these cultures. To begin, isolate whole spines from P5 to P10 mice and transfer them to a dissection microscope. With dissection scissors, cut through the ventral medial region of the spine in a longitudinal fashion. Carefully remove the dorsal root ganglia, or DRGs, with fine-tipped forceps. Transfer the ganglia to Hank's buffered salt solution in a petri dish. Trim any DRGs of their excessively long roots. Using a P1000 wide-bore pipette tip, transfer the ganglia to a 1.5 milliliter centrifuge tube. Keep the tube on ice until all mice have been processed. When DRG isolation is completed, centrifuge the tube at approximately 300 times gravity. Transfer the tube to a sterile tissue culture hood and remove the supernatant. Add 500 microliters of pre-warmed DRG papain to the tube and incubate in a 37-degree water bath for 10 minutes. When the papain digestion is complete, centrifuge, then replace the papain solution with 500 microliters of pre-warmed collagenase A solution. Incubate at 37 degrees for 10 minutes. Repeat the centrifugation step and replace the collagenase A solution with 1 milliliter of DRGN culture media. After a final centrifugation step, add 1 milliliter of DRGN media, and with a flame-polished glass Pasteur pipette, triturate the ganglia slowly at first, then with increasing intensity as the tissue begins to dissociate. Once no more tissue chunks are visible, transfer the cell suspension to a petri dish containing 7 milliliters of DRGN media. Incubate the dish in a 37-degree incubator at 8.5% CO2 for 1¼ hours. After this incubation, observe the petri dish under phase contrast. DRG neurons are evident as large-bodied cells. Many contaminating cells should be firmly adhered to the petri dish. Transfer the media to a 15 milliliter conical tube and rinse the dish with 4 milliliters of DRGN media to collect any remaining neurons. Centrifuge the conical tube at 300 times gravity for 5 minutes. Resuspend the resulting pellet in 500 microliters of DRGN media. Count the cells using a humocytometer. Be sure to only count the DRG neurons. Seed 30 to 50,000 neurons per 12 mm laminin-coated cover slip in 1 milliliter of DRGN media. Culture the cells in an 8.5% CO2 tissue culture incubator. The next morning, perform a full media change by replacing the DRGN media with OL media, supplemented with 10 micromolar FUD-R. Every second day subsequent to this, perform a 3/4 media change with FUD-R supplemented OL media. When examined under the phase contrast microscope, at day 1, neurons have begun to extend a few processes. At day 3, the neuronal processes have lengthened. At day 6, a neurite bed has developed, composed of many intermingled processes. At day 9, this neurite bed has increased in density and is ready for the reception of oligodendrocytes. By growing mixed brain cultures alongside the neuron cultures, oligodendrocyte precursor cells may be purified at day 9 of the neuron cultures. They can then be seeded onto the neurite beds by adding 50,000 cells to each well of a 24-well dish. At day 6 of co-culture, cells reminiscent of oligodendrocytes can be seen interacting with the DRG neurons. These cultures can now be fixed and processed for immunocytochemistry. When enriched oligodendrocyte cultures are labeled with antibodies against the oligodendrocyte markers Ng2, MAG, and MBP, immunologically positive cells are evidenced displaying stage-specific morphologies. These cultures originate as a population of Ng2 positive oligodendrocyte precursors and over the 6 days of differentiation produce MAG positive mature oligodendrocytes. When SDS PAGE is performed on lysates obtained from oligodendrocyte-enriched cultures, it can be seen that oligodendrocytes express CNP and MBP over the 6-day time course. When looking at DRG neurons prior to the addition of oligodendrocytes, dense TUJ1 and neurofilament-positive neurite beds are evidenced at day 9. At day 6 of oligodendrocyte DRG neuron co-culture, many MBP-positive oligodendrocytes can be seen interacting with the neuron neurites. At higher magnification, oligodendrocytes are seen to wrap these neurites with MBP-positive membrane, reminiscent of myelination. A digitally magnified view shows the extent of overlap of the MBP-positive membrane on the neurofilament-positive process.
Hi, I'm Scott Ryan, a postdoctoral fellow at the Ottawa Hospital Research Institute. These techniques provide a means of generating oligodendrocyte and DRG neuron co-culture systems from mice. This technique is valuable in studying both experimentally induced and transgenic models of myelinating disease. It is also effective for screening novel therapeutic compounds.